Okay, let's get set up. It actually, I can see with the camera's picking up this pretty good, so. No, pretty good. 75 kDA.
Our class is so focused on what cancer does and what cancer can do. However, cancer cannot cripple love.
Shatter hope.
It can't corrode faith.
It can't destroy peace.
And kill friendship.
It can't suppress memories.
It can't silence courage.
Can't invade soul.
And it can't steal eternal life.
So our treatment that we're going to be talking today is Novo TTF 100A, or Optane, which is used to treat glioblastoma.
So before we talk about the treatment, we're going to first talk about what it actually treats. Glioblastoma is a highly malignant, highly aggressive form of cancer in the brain. It has a very low median survival length. Without treatment, median survival length is about 4.5 months, and with treatment, it is around 14.5 months. Some common side effects are nausea, headaches, vomiting. It's more common in men than women, and it's more common in Caucasians and Hispanics, and we mostly see it in people over the age of 50.
We wanted to research glioblastoma because in my grandfather's early 70s, he was diagnosed with glioblastoma. He had surgery to have it removed, but unfortunately it had already metastasized and he died 2 months later. There's still a lot unknown about glioblastoma. As you can see, it has one of the lowest rates of survival and one of the lowest times for survival.
So Novo TTF 100A is a battery-powered device that produces alternating electric fields through transducer arrays, or just these pads you put directly on the skin. And these electric fields are also known as TT fields, or tumor treating fields. And the pads that you put on, the only known side effects are irritation to the skin and headaches, which they're a lot less severe than the normal side effects.
So Novo TTF, the electric fields produced by this device, they only target the cells when they're dividing, more specifically during mitosis at anaphase. And so basically how it works is it aligns structures in the cell, which then inhibit the division of the cell into two. And the two fundamental principles that allow for these electric fields to accomplish this are called dipole alignment and dielectrophoresis, which we will get into on the next slides.
So dipole alignment is the disruption of the formation of microtubules by electric fields being emitted into the cell. Electric fields are constantly switching so that the dipoles are constantly oscillating in sync with the field positive to negative. And at some point they move so fast that they just stay where they are and are unable to then form a microtubule, which leads to prolonged mitosis, metaphase arrest, and then cell death.
So dielectrophoresis is the uneven distribution of these electric fields during anaphase. As you can see here, this cell is in anaphase, about to split, and the electric fields, there's a high density of electric fields in the center, and there's a lower density on the outside. So this high density of electric fields, as you can see here, will attract the polarizable objects to the center, and this can lead to the uneven distribution of DNA or prolonged mitosis, which will both lead to apoptosis.
So one fundamental problem with this treatment is that the aforementioned principles, they only affect cells when the electric field is actually parallel to the direction of division. So as you can see in this picture, the electric fields do not affect the cells that are dividing here because the electric field is going left to right while the cell is dividing up and down. So the way that the researchers overcame this was they constantly switched the direction of the electric field. So 1 second, the electric field will be perpendicular to the cells here and will leave the cell unaffected, but then the next second, the electric field will switch directions, going parallel to the cell, and will then be able to target the maximum amount of cancer cells.
Are there any questions on the overall mechanism so far?
So it's just pretty much when the cell is in the phase, it's about to divide, and the waves or the field is coming over it. So in the center, there would be a higher density of fields, which would attract the polarizable object. So from the outside, it would come towards the center. And then if the cell was able to split, there would be an uneven distribution of DNA, or sometimes it would be able to split, and which would create a prolonged state of mitosis, and the cell would go through mitosis.
So are these two things pretty much doing the same thing, extending mitosis, the dipole alignment and the dielectrophoresis?
Dipole alignment is inhibiting microtubule formation, which then leads to prolonged mitosis and cell death. But in dipole alignment, also if the cells manage to split, it'll lead to uneven distribution of the DNA, which also leads to cell death. But dielectrophoresis is different because the microtubules are still forming and it's actually allowing the cell to split, but because of the electric field, it's causing all the polarizable objects to go to the center and then they won't split evenly.
Because dielectrophoresis won't occur until anaphase.
So your treatment, you want these processes to happen?
Yes. They only affect the dividing cells, and the cells, normal brain cells and neurons aren't normally divided.
So an experiment was done to test the efficacy of TT fields on cancer cells versus just cancer cells not being treated by anything. So as you can see, they both started at about the same time with about the same number of cells in the culture. And after 48 hours or 2 days, all the cells treated by TT fields that have attempted to divide have gone into apoptosis, while the cells that haven't attempted to divide are still alive. So there's much fewer cells in here than are in here because these cells have just grown uncontrollably, untreated by anything.
Okay, so next we're going to show you some pictures of some abnormalities that can occur as a result of these electric fields or TT fields. And just want to point out the important parts are the blue shows the chromosomes and the green shows the spindles.
So this diagram here, basically these 6 pictures just show 6 different types of mitotic abnormalities that are a direct cause of the TT field application. And basically you don't really have to worry about the specifics of them. They just show that the TT fields interrupt mitosis. And this graph over here just basically shows that over here on the right are the abnormalities, and over here on the left are the normal stages of mitosis. And as you can see on the right, the abnormal states of mitosis have a much lower percent of dividing cells. So that basically means that cells that are in this state of mitosis, these abnormal states, are dividing much less than the normal cells. The black is the with treatment, and the white is the control.
You don't worry about the specifics of the diagram. You just have to know abnormal states of mitosis caused by the TT fields are far fewer percent of dividing cells, while the normal states are much more dividing cells. So this graph shows Novo TTF versus the best physician's choice chemotherapy right now. And for the most part, it remains at a similar efficacy until about 24 months in here and about probably 20 weeks in here, where Novo TTF starts to show a little bit of a better efficacy than chemotherapy. But overall, they have very similar efficacy, but Novo TTF is far less toxic with less side effects and a greater quality of life for the patient.
Now we have some questions for you just to review what we've gone over. So can anyone tell us what type of fields are produced by the Novo TTF 100A?
Electric fields.
Through which two mechanisms does Novo TTF treat the tumor?
Dipole alignment and dielectrolysis.
Okay, and what structure does Novo TTF 100A inhibit formation?
And just as a sort of ending point, this graph here, it shows that glioblastoma, as in this picture, it has already been finished with its clinical trials and is FDA approved on, the most recent form of it was FDA approved on October 14, 2015. And, but the company who produces this treatment, Novo, is also looking to apply it to other cancers, for example, breast cancer, cervical cancer, all of these other different types of cancers. And treatment for those cancers are currently in the clinical trial stage.
So this treatment has a lot of potential with other forms of cancer.
Any questions?
What happens in regular cells when so when normal cells don't form microtubules, it can lead to diseases like Parkinson's, Rett syndrome, and a couple others.
What happens if the treatment is being done while the brain cells are divided? What will happen to those that are not?
Mature neurons do not divide in the brain. So they're unaffected.
Well, we don't really know to really talk about that, but
But brain cells in general don't divide. So only the glioblastoma cells within the brain will be dividing and then affected by electric.
Can you go back to the slide with the bar graph? The bar graph. So, you said that which bar is the one that was treated?
Black.
So, even when they're like only the abnormalities will only happen sometimes when you're taking treatment.
Yes, these are like, for example, when the cells are not like parallel to the field. If they're like perpendicular or dividing diagonally, then the TT field will have no effect on those cells.
Can you re-explain what the picture on the left shows?
So these 6 pictures just basically show different types of abnormalities to mitosis that are direct effects of the TT field application.
so, for example, this one is asymmetric anaphase. So which this pretty much means that one side of the cell, the membrane will be extended. So you can see here that this side of the membrane is kind of bigger than this side.
They're all just different types of abnormalities.
Yeah.
So they don't so they don't know which one of these abnormalities will happen?
It could, any one of these could happen to any.
Okay, so this chart on the right is basically saying that when cells are divided, they have abnormal mitotic formation. Then, if they're not treated with the drug, the cells will continue to divide.
No, it's basically just saying that when cells are in these states, these abnormal states as pictured here, they just don't divide because they don't divide as much as cells in normal states.
What's the y-axis on the chart on the right?
Percent dividing cells. So it basically means that of all of these types, or all of these states that mitosis is in, It's how many of those total cells, what percentage of those are actually dividing.
Is this specifically